Okay, so good morning, everyone. So today, me, Suffian, Rebecca, Sarah, and Tanjai will be presenting HIV and AIDS to you. Next. All right, next slide. Okay. So now we have the background on HIV and AIDS. In 1920, the first ever case of HIV was reported in Democratic Republic of Congo. In 1959, the HIV has been found in blood samples from several African nations. In 1981, AIDS appeared in the news with cases being reported in Los Angeles, United States of America. And in 1983, pathogen causing the disease had been identified. Okay, so now we have our learning outcomes. First, we will recognize the structure of human immunodeficiency virus and how it is transmitted. Secondly, we will identify the causative agent of acquired immunodeficiency disease. Thirdly, we will describe how HIV replicates and describe the treatments and preventions to control. Number 4, we will identify the symptoms of HIV and AIDS. Number 5, we will explain on the worldwide importance of HIV and AIDS. Number 6, we will explain on the differences between HIV and AIDS. Next slide, please. So now we will have a quick recap on immune system and immune response. So here's are the definitions of immune system. It is the body's internal defense systems. And the definitions of immune response is the complex series of responses of the body to the entry of a foreign and non-self antigen, which involve the activity of phagocyte and lymphocyte. There are two stages of immune response, non-specific immune response and specific immune response. Non-specific immune response. Phagocytic white blood cell engulf many foreign organisms, and in specific immune response, the specific clone of B and T lymphocytes can recognize specific antigens, then launch a specific immune response towards the specific antigen, which is primary and secondary. Now I'll pass it to Sarah for more information on HIV and AIDS.
Okay, so now I'll just briefly explain what is HIV. HIV stands for Human Immunodeficiency Virus. It belongs to a class of viruses called retrovirus. Retro means reverse. Once inside a host cell, the viral RNA is converted back to DNA to be incorporated into human chromosomes. So basically, this retrovirus, it works in a back to front way. How does it work? The retroviruses store their genetic information using RNA instead of DNA, meaning they need to make DNA when they enter a human cell in order to make new copies of themselves. Next, HIV cannot replicate on its own, and HIV infects the cells of the human immune system called the T helper cells. As you can see on so as you can see on the right top is the structure of the HIV virus. So as you can see, the two red strands are basically the RNA strands. The HIV virus is spherical. And then as you can see, the yellow area there, that is the capsid. The capsid is a protein coat. And then it is surrounded by the viral envelope, which is the outermost layer. It's composed of a lipid bilayer and glycoprotein. Okay, next slide. Okay, now what is AIDS? AIDS stands for Acquired Immunodeficiency Syndrome. AIDS is called acquired to distinguish it from other types, for example, an inherited form. So basically, it is called acquired because it is infectious and it cannot be inherited genetically. AIDS is a late stage of HIV infection, and if AIDS does develop, it means that the immune system is severely compromised and weakened, where it can no longer successfully respond against most infections and diseases. Now I'll pass it on to Rebecca.
So what are the causes of AIDS? AIDS is caused by a human immunodeficiency virus, also known as HIV. There are two types of HIV. First is HIV-1, which is more common and more transmissible, whereas HIV-2 is less common and less transmissible, as well as less responsive to drugs used to treat HIV-1. HIV has antigenic variation, whereby it can undergo mutation to change its antigen structure, so it can repeatedly change its protein codes, so it's very difficult for lymphocytes to recognize it and to remove it from our immune system. So what cells do HIV target? The HIV mainly targets the T helper lymphocytes, which plays an important role in activating our immune system. It also targets the macrophages and brain cells, such as the microglia cells. Next slide, please. So how does HIV cause AIDS exactly? First, the HIV virus will have to enter the internal environment of our body, which is inclusive of the blood, lymph, and interstitial fluid, and from that it infects the T helper lymphocytes. The virus uses the cell machinery of the T helper lymphocytes to replicate. The viral RNA, as you can see in number 1, the viral RNA binds to this glycoprotein known as CD4, which is also a co-receptor, and it enters the T helper lymphocytes through endocytosis, as you can see in number 2. Then as you follow the arrows, you can see that the viral reverse transcriptase enzyme uses RNA as a template to produce viral DNA copies, and the viral DNA copies are transported into the nucleus, where it is then inserted into the chromosomes. And during cell division, the viral DNA will transcribe for viral mRNA, which will translate for viral protein, which will produce more HIV particles. Next slide, please. So in the early stages of infection, infected cells remain relatively normal because viral DNA remains inactive in the cell, and this is known as latent virus. It can stay inactive in the cell for many years because HIV has antigenic concealment, whereby it can hide in the T helper lymphocytes without being detected by the immune system. At this point of time, the individual is HIV positive, you do not have AIDS yet. So after several years, the viral DNA becomes active and controls the cell machinery of the T helper lymphocytes. And it will produce more HIV particles, and this causes T helper lymphocytes to die. Why does it die exactly? It's because the viral reverse transcriptase enzyme is actually very ineffective and will code for incomplete viral DNA. And this will be detected by our immune system and will cause a programmed cell death known as pyroptosis, which is highly inflammatory. So when the T helper lymphocytes die, the number of T helper lymphocytes decreases and the number of lymphokines produced will decrease as well. So less B cells undergo cell selection and cell expansion, and less plasma cells are produced to secrete less antibodies. So the body becomes weakened to the point that it is unable to defend against infection. And this makes our body very vulnerable to many opportunistic infections, and this causes AIDS. So what are opportunistic infections? Opportunistic infections can be defined as infections caused by pathogens that take advantage of a host with a weakened immune system. So you can think of the pathogens as a bully and your immune system as a victim. So if you're a bully, you're most likely to bully someone who's weaker than you because they aren't able to put out any defenses. So the same thing goes with opportunistic infections. Pathogens will take advantage of you because you don't have any defenses against it. Next slide, please. So what are the symptoms of HIV infection? In the early stages of infection, HIV cannot be detected during the window period because antibodies are not yet produced. So HIV is a slow virus and it has a long incubation period. That means it takes a long time before you develop any symptoms. And when you develop symptoms, you will most likely develop a flu-like symptoms, which will last for several weeks. Then you become symptomless for a few months to around 10 years because the virus is inactive in the host cell. So at this point of time, the individual is HIV positive and do not have AIDS yet. Next slide, please. So what are the symptoms of AIDS? So when you have AIDS, you will get opportunistic infections by fungus and by bacteria. So opportunistic infections by fungus are such as oral thrush caused by Candida albicans. As you can see in the corner, that is oral thrush. And you get pneumonia as well, which is caused by Pneumocystis jirovecii. Then opportunistic infections by bacteria are such as tuberculosis caused by Mycobacterium tuberculosis. When you have AIDS, you're more likely to get dementia because the HIV virus actually infects the microglia cells and the macrophages of the brain. Microglia cells are basically the macrophages of the brain and act as immune cells to create an optimum environment for your neurons. Next is when you have AIDS, you're more likely to get cancer as well because the immune system collapses to the point that it is less effective in detecting and destroying cancer cells. Example of cancer is a rare form of skin cancer known as Kaposi sarcoma caused by herpes-like virus, and there is also cancers caused by cancers of internal organs. Other symptoms are such as fever, weight loss, diarrhea, and sweating. Next slide, please. So as you can see in the first picture, it is pneumocystis pneumonia, and you can see those white stuff around it. This is caused by subtle consolidation, whereby there's a buildup of fluid in the lungs, such as buildup of like pus or blood. And you can see in normal lungs, you don't have that. And when you have tuberculosis, when you develop tuberculosis because of AIDS, you're most likely to develop post-primary tuberculosis, which infects the upper lobes of the lung. As you can see here, can you tap the slide again? Yeah, as you can see here, the hilar structure is the one that is highlighted in yellow. In the normal lungs, the right hilar is lower than the left hilar, but when you have tuberculosis, it's around the same height because of a decreased volume in the upper lobe of the lung. And there's also apical consolidation where there's a buildup of fluid in the upper lobe of the lung. Next slide, please. And I'll pass it on to Suffian.
So on this slide, I'll be talking about how HIV is transmitted. So there are many ways HIV can be transmitted. HIV transmission is when HIV enters the body through direct body fluids or injecting infected cells. Most commonly, HIV is transmitted by having unprotected sex with an infected partner. The virus may enter the body through the genitals during sexual intercourse. Also, drug addicts are highly susceptible to HIV infection because HIV frequently spreads among injection with usage of unsterilized needles or syringes that are contaminated with blood from another HIV positive person. HIV may also spread through transfusion of contaminated blood during blood donation. To minimize the risk of infection, all blood products are to be tested for HIV and can be heat treated to kill any viruses. HIV can be transmitted from mothers to infants during pregnancy or childbirth through the placenta. Mother-to-child transmission can also occur through breastfeeding, as both viral particles and infected lymphocytes are found in the breast milk. HIV can also spread in healthcare settings through accidental needle sticks or contact with contaminated fluids or through non-sterile instruments. However, these days there are misconceptions and misunderstandings of how HIV can be transmitted. So as you can see, HIV is not transmitted through the sharing of clothes, personal items, sharing food and drinks, insect bites, household items, and coughing and sneezing. So because HIV, it can't live outside the body, so you're safe. Next slide, please.
Okay, so HIV can be diagnosed through blood or saliva testing. There are 3 tests, antigen test, antibody test, and nucleic acid test, which is known as NAT. The antigen test, the test usually involved by drawing blood from a vein, and the antigens are substances on the HIV virus itself and are usually detectable. The antibody test, the test look for antibodies to HIV in our blood or saliva. It is the most rapid HIV test as it includes self-tests that can be done at home. The NAT, the nucleic acid test, the test look for the actual virus in your blood. They also involve blood drawn from a vein. So if you might have been exposed to HIV within the past few weeks, your doctor may recommend NAT. NAT will be the first test to become positive after exposure to HIV. If any of the tests are negative, you may still need a follow-up test week 2 months later to confirm the results. Next slide, please.
So how can HIV be prevented? First is to restrain from sharing needles among drug users. The reason why drug users even share needles is because it's very difficult to acquire needles without a prescription. So to combat this, needle exchange schemes are implemented so that you can exchange your needles for sterile ones. Next is to screen blood donors for HIV. Nowadays, many people donate their own blood and store it in blood banks in case of an emergency or to prepare for an operation. You can also heat treat blood products because HIV virus can only survive in temperatures around our body temperature, and under high temperature, it will undergo denaturation. Third is to use contact tracing to practice. Contact tracing is the practice of identifying and informing someone that a partner they had sexual intercourse with or shared syringes with is HIV positive. So they will get them to take the HIV test to identify the presence of antibodies to HIV. The HIV test is best tested after 3 weeks after initial infection because there's a window period whereby you haven't produced antibodies against this virus yet. So it's very difficult to get a more accurate result. It's best to be tested after 3 weeks. 4 is to do widespread testing of the population, and 5 is to abstain from breastfeeding if you're a HIV positive woman in a high income country. Because if you're a HIV positive woman in a low income or middle income country and you have access to drugs such as the antiretroviral drug, it's best to continue breastfeeding because you may not have access to clean water to mix the formula milk with to feed your baby. Next slide, please. Next is to take the antiretroviral drug if you're sexually active or if you're a pregnant woman. If you're a pregnant woman, the antiretroviral drugs prevents the vertical transmission of HIV from mother to fetus through the placenta. It also prevents the transmission of HIV to baby during delivery or breastfeeding. It also reduces the risk of sexually transmitting HIV to HIV-negative partners. It's also best to use a condom, femidom, or dental dams when having sexual intercourse. A dental dam is basically this thing you can see in the picture, where you apply on the outer part of the genitals or on the anus during oral sex. So it forms a barrier between body fluids, reducing the chances of transmission of HIV. Next is to practice monogamy, which is basically having only one partner. Next is public health measures, where you educate people about the spread of infections and encourage people to change their behaviors to protect themselves and others. Lastly is to organize campaigns for HIV and AIDS to once again educate people about the spread of infection.
So there is yet no cure for AIDS and no vaccine for HIV. However, HIV positive people are treated with drugs so they can live with HIV. Drugs therapy can slow down the onset of AIDS so that many HIV positive people has a longer life expectancy. However, drugs are expensive and have a wide range of side effects ranging from mild and temporary, such as rashes, headaches, and diarrhea, to the severe and permanent, such as nerve damage and abnormal fat distribution. Next slide, please. So over the past years, many drugs have been developed to combat HIV and its associated infections. These drugs are called highly active antiretroviral therapy, which is known as HAART, and these drugs have significantly played its role in reducing HIV complications, HIV-related complications, and thus avoiding deaths. So the drugs are similar to DNA nucleotides. So over here, there are 5 main drug classes: fusion inhibitors, nucleoside or non-nucleoside reverse transcriptase inhibitors, protease inhibitors, CCR5 antagonists, and integrase inhibitors. So on the right is the mechanism for each of the drug classes. So for fusion inhibitors, it blocks HIV from fusing with a cell's membrane. And non-nucleoside, their main mechanism is preventing conversion of RNA to DNA. For protease inhibitors, they break HIV protein chain into parts. And CCR5 antagonists, they stop HIV from entering uninfected cells. And integrase inhibitors, they prevent HIV from being incorporated into DNA. So these drugs, however, do not cure HIV or AIDS. Next slide. So for example, we have the Zydovidin. Zydovidin is similar to the nucleotide that contains the base thymine. So Zydovidin is in a class of medications called nucleoside reverse transcriptase inhibitors, or for short, it is known as NRTIs. So to summarize the mechanism for Zydovidin, it basically binds to the viral enzyme transcriptase, which blocks its action and prevent replication of the viral genetic material. And this helps to increase in some of the body's lymphocytes. The pattern and timing of medication through the day must be strictly followed. People who are unable to keep to this pattern can become susceptible to strains of HIV that have developed resistance. Next slide, please. So over here, you can see a diagram of Zydovidin. Zydovidin in short is known as AZT. So over here, I'll explain. When HIV is in a T cell, it releases enzymes with the HIV genetic material. Viral enzymes released into the cytoplasm leads to the activation of reverse transcriptase enzyme. So on the diagram, the RT, the cuboidal RT here is the enzyme. The conversion of, so once this enzyme is activated, the conversion of viral RNA into single-stranded DNA can begin. So the reverse transcriptase enzyme moves along HIV RNA molecule and copies it into viral DNA through the incorporation of naturally occurring nucleosides. So AZT compete with the natural nucleosides to prevent incorporation into viral DNA and thus prevents reverse transcription. So RT, the reverse transcriptase, it embeds the AZT into synthesized molecule of DNA, and as a consequence, natural nucleosides can no longer be added. So this process is called as chain termination. Next slide, please. So what if a HIV positive person refused to be treated? When a person contracts HIV and if they do not receive treatment, the infection will typically progress through 3 stages. The first stage is known as acute infection. So soon after contracting the virus, the person has a large amount of HIV in their blood, and the risk of the virus transmitting to others is high. Some people may experience flu-like symptoms, but others will have no symptoms. The second stage is called chronic infection. So without treatment, this stage can last for a decade or longer. People may not have any symptoms, but the virus can still transmit to other people. So the last stage is known as AIDS. At this stage, a person's immune system is badly damaged and their T cell count drops below 200 cells per cubic millimeter of blood compared to normal cell count, which is 500 to 1,600 cells per cubic millimeter. So they will become vulnerable to opportunistic infections, and the risk of viral transmission is high.
Okay, so now let's discuss the differences between HIV and AIDS. So HIV, for one, is a virus, a small infectious agent that multiplies itself by taking control of cells inside a host. AIDS, on the other hand, is a syndrome, a group of connected symptoms that are usually caused by a single disease or virus. Next, HIV can be transmitted from person to person and also damages the immune system. AIDS results when HIV damages the human immune system so severely that the person becomes very susceptible to additional problems, including infections like pneumonia and tuberculosis. Lastly, HIV should only refer to human immunodeficiency viruses, and AIDS should only refer to the relatively end-stage syndromes after HIV has extensively damaged a person's immune system. So, for example, a person can have HIV, or better termed an infection caused by the human immunodeficiency virus, but not have AIDS. Next slide.
All right, so now we have the estimated number of global infected people with HIV. In 2004, more than half of the people living with HIV were female. In 2019, 38 million were adults and 1.8 million were children. And in 2020, 35.9 million were adults and 1.7 million were children. Next slide, please. So as you can see in this diagram, we will focus on the Sub-Saharan Africa, where the number is highlighted in red, which is 28.5 million. It is because of Sub-Saharan Africa consists of less developed countries, which means that they have a picture of low per income capita and widespread poverty. Poverty enhanced the spread of HIV and AIDS. So lack of knowledge means like they have restricted amount of information on the disease. This is due to having less resources and less campaigns made. Also, they have lack of accessibility to healthcare services, which it causes the HIV infection to make people more vulnerable to existing diseases such as malaria and malnutrition. Next slide, please. So here we have a comparison, which is the global HIV and AIDS epidemic, adult HIV prevalence in 2019. Epidemic means the rapid spread of disease to a large number of people in a given population within a short period of time. And prevalence, it means the total number of individuals, a population who have who have disease or health condition at a specific period of time. They are usually expressed as a percentage of the population. So if you can see the yellow key, which is marked under the southern part of Africa, it consists of 8 countries, such as Namibia, Botswana, Lesotho, Zimbabwe, Mozambique, Swaziland, Zambia, and also South Africa. So these countries is considered to be the more than 10% contribution to the global prevalence, which is 0.7%. So now we will discuss on the worldwide importance of HIV and AIDS and the factors that influence its global pattern distribution. So number 1, it promotes opportunistic infection, such as tuberculosis, pandemic, malaria, and pneumonia. Secondly, economical loss. It affects people in their 20s and 30s who are most economically productive and sexually active. It also, the expensive drugs also drain the government funds. Number 3, no cure for AIDS. The drug therapy only slows down the onset of AIDS and hence prolongs life. Drugs are expensive and they have mild to severe side effects. Number 4, no vaccine for HIV. HIV often changes its surface proteins, so hard for immune system to recognize it. Number 5, the long lantern stage. The carrier may be infected for many years and transmit HIV to others without knowing that he is HIV positive. Number 6, the HIV infection cannot be detected during the window period, which is the several weeks after the infection when the antibodies are not produced yet. So number 7, in Africa and Southeast Asia, the disease is widespread beyond the high-risk groups. This is because of the difficulties of reaching the majority of population and organized testing. I'll pass it on to Sarah.
Okay, so now let's look at this question 4 from October-November 28. Suggest and explain the global importance of providing ART to all pregnant and breastfeeding women living with HIV. So first of all, we have to, ART is antiretroviral therapy, and we already know that ART also involves taking a combination of HIV medicines. So from earlier, we already know how ART helps women in general, especially pregnant women. It prevents vertical transmission of HIV from mother to fetus through the placenta and prevents transmission of HIV to baby during delivery or breastfeeding. So for the first point, we can talk about how HIV can be vertically transmitted from mother to baby. Therefore, by providing the antiretroviral therapy, it can decrease the HIV transmission during pregnancy, like through the placenta, during labor and breastfeeding. So this reduces the number of babies born with HIV, so lesser death rate. For number 2, we can talk about how ART reduces the number of HIV positive women becoming ill with opportunistic infections like tuberculosis and pneumonia. Basically, opportunistic infections is caused by, like, when you have HIV, your immune system is severely compromised and weak, so it's harder for your body to fight off infections. And for point 3, by providing ART, we can reduce the spread of HIV as well. So for example, by providing ART to the pregnant and breastfeeding mother, the child may grow up without HIV and will not pass on, and the mother is also less likely to pass to the partner. And for the last point, we can talk about the social and economic effects. So by providing ART to HIV positive women, they can live a more healthy life so they can contribute to workforce. And it can also be their main career if their partner has died. If infants are not born with HIV, then no lifelong ART is required, and it makes breastfeeding safer when no other options exist to feed babies. Next slide. So for this next question is from May-June 2019. People with HIV and AIDS have a low helper T lymphocyte count. Explain how a low helper T lymphocyte count could reduce the body's ability to produce antibodies against Vibrio cholerae. So first, we have to know how HIV and AIDS cause a low helper T lymphocyte count. Basically, the HIV enters its genetic information into the helper T cells to make copies of itself. So when this happens, the helper T lymphocytes die, which disrupts the immune system. So for the first point, we can talk about how a low helper T lymphocyte count releases lesser cytokines. Then after that, the other point, we can talk about any one of this. So we can talk about how fewer B lymphocytes divide by mitosis, or fewer B lymphocytes stimulated to differentiate into plasma cells, or fewer plasma cells to produce and secrete antibodies. Next slide. Okay, so this is the summary chart. HIV stands for human immunodeficiency virus. It is a retrovirus that attacks the human immune system. So earlier we have already seen the structure of the HIV virus. So we know it is spherical. It is composed of 2 RNA strands. There is a protein coat called the capsid. The viral envelope, which is the outermost layer, it is a lipid, bilayer, and glycoprotein and attachment protein. And I'll pass it on to Rebecca.
So we're now going to talk about the replication of HIV. The HIV will have to attach to the co-receptor on the T helper lymphocytes, and the virus first enters the bloodstream and infects the T helper lymphocytes. Then the single-stranded RNA is converted to double-stranded DNA by the reverse transcriptase enzyme. The viral DNA copy is then inserted into the chromosome of the T helper cells. The infected cells remain relatively normal as viral DNA is inactive. Then the budding process. The viral DNA within the host cells become active, then it takes control of the cell machinery of the T helper cells. This causes more HIV particles to be produced and cause T helper lymphocytes to die. Thousands of HIV particles are released and able to infect other T helper cells. So how does HIV affect the immune system? As the HIV reduces the number of T helper lymphocytes, less B cells undergo cell expansion and cell selection at cell expansion to produce less plasma cells, and the number of antibodies decreases. So this decreases the body's ability to fight off infections, leading to AIDS, which is acquired immunodeficiency syndrome. AIDS is a collection of opportunistic diseases associated with immunodeficiency caused by HIV. Now I'll pass it on to SUFFIAN. Oh wait, no, it's me. So what are the symptoms of HIV positive? What happens when you're HIV positive? You'll first develop flu-like symptoms, which will last for several weeks. Symptoms of AIDS are such as pneumonia, tuberculosis, dementia, and lung cancer. And now I'll pass it on to SUFFIAN.
So how HIV transmitted? It is a direct exchange of body fluids. So first, it's through sexual intercourse, blood donation, sharing needles used by intravenous drug users, from mother to child through breast milk, and from mother to child across the placenta. So in treatment, there is yet no cure for AIDS and no vaccine for HIV. So HIV cannot be cured. Drugs therapy, however, it can slow down the onset of AIDS so that many HIV positive people have a longer life expectancy.
So how do you prevent HIV or AIDS? First is to not share syringes when you're taking drugs, and to use protection such as condoms, famidoms, and dental dams, and to get tested. Thank you. That's all from us. Oh wait, no, the video.